pure culture is crucial for accurate identification and further study of microorganisms, and streaking method is essential for obtaining a pure culture from a mixed population of microorganisms. There are several types of streaking methods used to isolate pure bacterial colonies on solid media. The main streaking types are quadrant streak method, T-streak method, radiant streak method, Zig-Zag streak method, and continuous streak method. Today, we will learn how to perform Zig-Zag streaking method or technique, which is ideal for checking viability, preparing bacterial lawns, or conducting antimicrobial sensitivity tests. Welcome to this video. Let's get started. For streaking techniques, an inoculating loop is essential. It typically consists of a nichrome or platinum wire attached to a handle. In some cases, pre-sterilized disposable plastic loops are used. A Bunsen burner is also required for sterilizing the loop before and after use. To cultivate microbes, we need appropriate culture media plates, such as nutrient agar or selective media. Additionally, a mixed microbial culture plate is needed as the source from which we aim to isolate a pure culture. On the mixed culture plate, you can observe colonies of various microorganisms, differing in shape, size, and color, indicating the presence of multiple species. This is another type of plate that contains a single type of colony, which can also serve as a microbial source. Let's perform zigzag streaking. First of all, flame and burn the loop in the Bunsen burner until red hot. Let the loop cool for a few seconds. Gently touch and pick a single, well-isolated colony from the mixed microbial culture plate using a sterile loop. Prepare a smear using the bacterial cells collected on the loop on the edge of the media surface. Flame the loop in the Bunsen burner until red hot and let it cool for a few seconds. Starting from a distance edge, draw a continuous zigzag line across the agar surface, touching the smear from one side to the other. In this way, continue zigzag from the top to the bottom of the plate. This type of streaking method is relatively less effective to obtain isolated colony. Close the lid of the Petri dish, invert the plate, and incubate it at 35 to 37 degrees Celsius for 24 to 48 hours to allow optimal bacterial growth. After incubation, carefully remove the plate from the incubator and prepare to observe the results. In the zigzag streaking plate, you can see a continuous line of bacterial growth following the zigzag path across the agar surface. Since this method does not involve dilution between streaks, isolated colonies are very less. Few isolated colonies are observed at the bottom of the plate. This type of streaking is ideal for checking viability, preparing bacterial lawns, or conducting antimicrobial sensitivity tests,